It's really a pleasure to be back in Guelph and to be back with CCSAW. And congratulations to the animal judging teams, wherever you may be in the room or online. So today I'll be telling you about the research program that we put in place at Université Laval on swine behavior and welfare. And I'll be telling you essentially about three main projects that are all united by a central concept, which is robustness. Just want to check. The people in the back of the room, can you hear me properly? All right. Thank you. So what is robustness? We're defining robustness here as the capacity of an animal to face challenges to its welfare and to be minimally affected by them. To give you a sort of conceptual framework for robustness, I'm going to start with a version of a diagram that you may have seen before from Fraser and Company, which represents on the left side in that first circle, the things that an animal is able to do, the types of challenges that animal is equipped to face as a function of its current capacities. And on the right side, we have the things that the animal's environment requires the animal to do in order to maintain good welfare. When those circles overlap, things tend to go very well, though not perfect necessarily, but welfare problems tend to emerge when there are zones of non-overlap between the two circles, when there are challenges, for example, the environment poses to the animal that the animal is not equipped to face. And most efforts to improve welfare involve modifying the environment to fit it better to the animal. So if you think, for example, about providing animals with environmental enrichment or about making their environments more naturalistic, these are efforts to fit the environment to the animal, to make the environment present challenges that the animal knows how to solve. And this is a very important approach. It's fundamental. In fact, you'll see that it features into all of the projects that I'll discuss today. But really we'll be focused on what I consider to be a complementary approach, which is finding ways to make the animal better fit the environment. In other words, giving the animal the tools to overcome the challenges that the environment poses. And in many cases, these are challenges that we're not necessarily able to avoid presenting to the animal. So I'll be telling you about three main types of potential determinants of robustness that we are investigating in our research at Laval. Before I start talking about these projects, I should stress two things. Number one, these projects are all at various different stages of advancement. So what I'm going to present today is neither complete nor final. Some of it is data that came in just last week, less than 7 days ago. So this is a work in progress. And I should additionally stress, I have 45 minutes to present today. 2 days ago, I had 100 minutes of content. So I'm going to give you the big picture, and I'd be happy to fill in some details during question period or if we chat later on. So I'm going to tell you a little bit about the context where we're working. We're working on pigs. Why pigs? Well, there are several reasons to be interested in swine for this research. One is that simply from an applied welfare perspective, the swine industry is huge. Canada is one of the biggest producers internationally. We produce over 25 million swine per year in Canada. And Quebec in particular, where we're situated, is the largest producing province. So for that reason, research on pig welfare has the potential to have a widespread impact on welfare if it leads to changes being adopted in the industry. In terms of robustness, we're also interested in swine because this is a species that faces multiple challenges to its welfare. And some of these challenges we can try to optimize the environment, but the challenges are not necessarily things that are going to go away entirely. Some of the big challenges to swine welfare include, for example, pathogenic risk. So if you compare the swine industry to some of the other big animal production industries, they tend to be quite paranoid about biosecurity, and with good reason. Respiratory diseases in particular are the leading cause of post-weaning mortality in pigs. They account for quite a high percentage of mortality. And the typical response in the past has been to use antibiotics to prevent these diseases from setting in. But of course, there are concerns about antibiotic resistance. And so we're looking for other ways of making sure that pigs are not so vulnerable to this pathogenic risk, which does creep into farms despite strict biosecurity measures. We can think of other welfare issues that are maybe particular to the pig industry. One of them is confinement and single housing of animals. If we think particularly of breeding sows at various stages of the production cycle, they are housed by themselves, which is not the most appropriate type of housing necessarily for a gregarious species. We can think about other challenges like weaning, for example, and every other transition between stages of production where animals face various challenges, including being mixed with new groups of animals, and injurious aggression is one of the particular challenges that can be quite severe in swine. And then, of course, as in any meat production sector, we have to worry about human handling, interaction, stress at transport to slaughter, which is an inevitable aspect of our current production system. So these are types of challenges that we're not going to make go away tomorrow by modifying the environment. And so that's where the interest in modifying the pig to better be able to overcome these challenges comes in. The logos you see on the bottom are the logos of the two organizations we work with where we do the research. On the left, CRSAD, which is pronounced CSAD. This is the Provincial Animal Science Experimental Farm, which has multiple sectors of production on site. It's about 45 minutes away from Quebec City, where we are. And they have 2 experimental piglet nurseries and 1 grow finish unit on site. And on the right, we have the CDPQ, the Quebec Pork Development Center. This is a public research and knowledge transfer organization, which is dedicated to the Quebec swine sector. And they have 1 experimental sow unit and then 1 nursery grow finish unit. And I'm going to tell you about research that happens in essentially all of these buildings today. So let's start with our first potential determinant of robustness with respect to pig welfare. Here we're going to think about personality and genetics. We're going to think about spontaneous differences between individuals in their predisposition to maintain either good welfare or bad welfare. I'm just showing you here a sample of some of the titles of documents that I was writing very early on when I got this job at Laval a few years ago. The titles are a little bit provocative, but they kind of convey the thought that I'm about to explain in more detail, which is that well-being or welfare is a phenotype that we should select for. Where does this idea come from? It comes from psychological research on humans, where some researchers study something they call subjective well-being. It's a little bit like what we study in animals, right? And the way that they typically measure this is by asking people to self-report. So they'll say, how do you feel? How happy are you on a scale of 1 to 10? How would you rate your quality of life on a scale of 1 to 10? And this kind of research teaches us some very interesting things. One is that differences between individuals tend to be pretty stable over time. So someone who tells you 8 out of 10 today, you ask them again in 3 years, they'll usually give you an answer that's pretty close to 8 out of 10. Someone who's a 2 out of 10, they'll give you an answer close to 2 out of 10 in a few years again. These differences are also surprisingly insensitive to external circumstances. So if you look at factors that we would think would have a big impact on quality of life, like family status or income, they don't actually have as much of an impact as we would think, outside, of course, of crushing poverty levels. They're also relatively insensitive to perturbations. So you have studies on people who lose their spouse, okay, their spouse dies, they lose their job, they are in a car accident and they become paraplegic, or on the other hand, they win the lottery, and that's going to cause a big dip or a big rise in their subjective well-being, but that's going to last maybe a year, 2 years, and eventually they adapt to their circumstances and they come back generally to something close to their baseline level, whether that's an 8 or whether that's a 2. You put this together, and that tells us that subjective well-being in humans is something that we can consider robust because it doesn't seem to be that affected by external challenges. And it seems like it's an aspect of the individual's personality because it results not just from external circumstances, but from how people perceive those circumstances and interpret them. The last thing that this research tells us, research on genetics and genomics in humans, is that subjective well-being seems to be at least partially heritable. Which brings up the idea, what if things work this way in animals too? Could we select animals for good welfare, more or less regardless of the circumstances the animals find themselves in? So the project that this leads to, of course, is that we're going to recruit a bunch of pigs. We will ask them, scale of 1 to 10, how do you feel? We'll breed together the ones who give us big numbers, then we'll interview their kids, see if they give us a big number too. Obviously, we can't. I'm a little bit jealous of the psychologists because I work with nonverbal animals. So how do we actually measure subjective well-being in animals? I think the closest we can get to something like subjective well-being is using a family of methods of measurement of welfare assessment called cognitive biases. And to make a very long story short, essentially this comes from, once again, work on human psychology, which recognizes that our underlying mood states, positive moods, negative moods, have an impact on our cognition, and our cognition has an impact on our behavior, which can be measured. And the best known type of cognitive bias that has been used in animal research and that has been validated as a welfare assessment across a wide range of different species, including pigs, is judgment bias. Essentially, judgment bias is the fact that if you are feeling in a positive mood, you tend to make optimistic judgments in a situation of uncertainty. Whereas if I don't actually need this mic, so I'll just unclip it. Whereas if you're feeling in a negative mood state, such as depression, for example, you tend to make pessimistic judgments in ambiguous situations. The way that we test this in animals generally is that we first teach them some sort of operant discrimination. So here I've represented this by a stimulus, which is a black circle, for example, which we're going to teach the animal to associate with a reward if they pose a certain action when they see the black circle. On the other hand, we teach them that if we show you a different stimulus, here a white circle, and you pose that same action, you will get no reward, or you might even get punished. So the animal learns to react differently when they see the black circle, which is a good sign for them, and the white circle, which is a bad sign for them. Once they've learned this discrimination, here's the test. You show them gray circle, and you ask them, does this look like a glass half full? Will you react to this like the black circle, showing us an optimistic response in front of uncertainty, which betrays your positive mood? Or will you treat this glass half empty, like a white circle, avoid taking the action, which tells us you're pessimistic and more likely in a negative mood state? Now, the thing is, if we want to use some sort of test like this for genetic selection, we're going to need to get data from lots of animals. When I started reading on quantitative genetics, when I got interested in this idea, my jaw dropped to the floor because I realized that people who do these kinds of selection experiments don't work with 20, 40, 60 animals. They work with hundreds upon hundreds of animals. And getting judgment bias measures is very labor intensive when you're training animals manually. And so this brought us to the first objective of the project that I'm presenting here, which has been done by PhD student Colonel K.L. And our first objective was to develop an apparatus to collect this type of data automatically. And what we came up with after some pilot projects and much refinements of the apparatus was what we could call an arcade cabinet for pigs. So this is a robust plastic device. Excuse me. I'd like to point with my mouse. There we go. Which can be installed in the home pens of piglets. So these are group house piglets, housed in groups of 4. And this goes into the, where did my mouse go? This goes into the corner of the pen. And when piglets enter this chamber here, their presence is detected. They are identified by an RFID tag on their ear. And the presence of the piglet initiates a trial. And then depending which piglet we're dealing with, which program we've put that piglet on, they'll be presented with either an auditory stimulus, which could be high-pitched or low-pitched. So that's equivalent to our black circle and white circle in the example from earlier. Or they could be presented with a visual stimulus on a computer screen, which is going to be gradients of colors. And now we're trying to teach the animal that discrimination, much like in what I presented earlier. And the animal can respond by pressing on joysticks over here, and correct responses will yield food rewards that are dispensed by a mechanism that looks like this. This is a bit of a duct-taped prototype version. When they give inappropriate responses, they can be punished using a timeout, which basically blocks them from trying again for a certain period of time. Now, this apparatus works well, and it has a big advantage over manual training, which is that if we look here, and keep in mind this is a logarithmic scale on the y-axis, the number of trials that we can obtain, which are voluntarily initiated by piglets going to the apparatus themselves in their home pens, is much greater. It's about 2 to 3 times greater every week than the maximum number of trials that Gwen up here, the PhD student, could train the same number of piglets on in 1 week if she was working about 6 hours a day, 5 days a week. And this is something that she's tried. So that's based on some real data. So we get a lot of data from this approach, which is great. However, it does have some problems. One is that the number of trials per individual can be quite variable. So there are a small percentage of individuals who pretty much don't take part in this test voluntarily. The other problems are that interest seems to decline over time. So this is the number of trials initiated per piglet per hour over the weeks of the test. Why is this happening? Partly they may be habituating to the reward, starting to get tired of that food reward. Partly they may also be reacting to the test itself getting harder as they progress through the stages of training. The other big problem, likely the biggest problem and maybe related to the first, is how trials end. The blue slice here representing maybe 37% of trials is when piglets initiate a test and then give us a response by pressing on one of the joysticks. And I did this, but they're actually using their snout. Then there's a small sliver of tests that time out because the piglet walks in, trial starts, the piglet leaves. But by far the thing that happens the most is interruptions by other pigs. They're always in each other's business. And this despite the fact that we made some tweaks between batches. So this is an earlier version here. This is the version from our last batch. We had added a little wall here that we can slide and fix in place so that we could make it this thin when the piglets are young and then make it a little bit wider when they're older. So only one could enter at a time. They still find a way to climb on top of each other, underneath each other, and get in each other's way. So we're getting a lot of data. We do have some problems, but the big question is, do the piglets learn? I didn't make a graph because it hurts my heart so much. They did not learn the operant discrimination, which means we could not run the automated judgment bias test here. We did, however, have a second objective in this project, which was related back to the concept of robustness. We want to see whether individual differences in welfare are stable over time and whether they're robust to changes in the animal's circumstances or changes in the animal's environment. So the way that we meant to evaluate this is to look at animals over time, take measures longitudinally, and we would evaluate stability in animals who stay in the same type of housing, which is represented here by the shading of the pens. And we would evaluate robustness in animals who midway through the nursery period, which I don't know if I've said this, but it's a 5-week period between weaning at 3 weeks and exit at about 8 weeks, piglets who would be switched from one type of housing to another type of housing. And the type of housing that we use, or the one I'm going to talk about today, is environmental enrichment. And we chose this because previous research had shown that it has impacts on different welfare indicators, including judgment bias measures. So generally, the enriched piglets are showing more optimistic responses in previous work. And so half of the piglets in the study were enriched with manipulable objects, as well as a secondary feeder, which was filled with peat moss, which they like to eat and they like to root around in. So it's a substrate that they're interested in. And thankfully, because we thought maybe the judgment bias task might not work, we collected data on complementary welfare indicators. So we were still able to evaluate stability and robustness of welfare according to these other indicators. One of the indicators we were interested in is hormonal, glucocorticoid hormones that reflect stress. And in particular, we wanted to see the ratio of cortisol to DHEA, which is, these are both glucocorticoid hormones, and this ratio is thought to reflect chronic stress. And previous work, this paper here by Fels et al., has shown that enriched piglets generally have a lower cortisol to DHEA ratio than standard housed piglets. We collected samples from the animals, saliva samples from the animals, and we did this on a voluntary basis in a low stress manner. So essentially this means that prior to the dates of sample collection, we had some sessions of just habituation to the piglets. And so by the time we wanted the real samples, the piglets were very much willing to just come up to us and munch on the cotton swabs, which we used to collect the samples. I'm not really going to show you the treatment differences so much because I want to focus on the stability and robustness of these measures. I'm just going to mention that cortisol itself, not the cortisol DHEA ratio, but cortisol itself was higher in the non-enriched animals than in the enriched animals. Actually, I do have the graph. Sorry about that. I thought I had removed it. Now, before I talk about stability and robustness, I want to make a little bit of a statistical detour to tell you how we're actually testing for this. When we want to know, is a measure stable? That means, first of all, we have to measure it twice, which we did for these hormonal assays at the middle of the testing nursery period, so at about 5.5 weeks of age, and again at the end of the testing period, at about 8 weeks of age. And if we want to see if this measure is stable, we are essentially looking for a correlation between the outcome of the first measurement and the outcome of the second measurement. The data on screen is not the data for the cortisol, for any of the hormonal indicators. It's from something else. It's just to illustrate the concept. Here we see a measure that looks quite stable because high values over on the right on the first test correlate with high values for the same animal on the second test. And meanwhile, on the left, animals who score low on the first test score low on the second test. This would be great evidence of stability, except we have to factor in other things like batch effects. So in this version of the graph, the color reflects the batch of the animal, a batch being a group of animals who come in all at the same time and who leave the experiment at the same time. Here we see that much of this correlation is actually not between individuals within batches, but actually between batches. And we could break this down further by taking into account pen effects, given that individuals within a same pen are not independent of each other. So if we take this same data and now we break it down by batch and by pen, now we're able to see, is there a positive correlation within each of these pens? And for this measure in particular, there does tend to be. How we're testing for stability statistically then is that we're running mixed models where we put the outcome of the second measurement as the thing we're trying to predict, and the outcome of the first measurement as one of our predictive factors. And we add to this as random factors, the batch effect and the pen nested within the batch. And on top of this, we also put in factors like, for example, housing treatment, which we think might influence these measures. And if we still, with all these controls in place, find a positive correlation between the first and second measurement, that gives us evidence that we have a measure that's stable in terms of differences between individuals across time and differences between pens that can't be explained by things like enrichment or batch effects. So running these types of analyses, what did we find for our hormonal indicators? We found that cortisol itself, just baseline cortisol levels, are not stable over time, which makes sense because they fluctuate a lot throughout the day. Whereas the concentrations of DHEA and to some extent, the ratio of cortisol to DHEA, which are thought to reflect chronic stress states, are more stable over time. This is evaluated stability in our wider sample. And if we want to evaluate robustness itself, now we need to look at this same analysis within a subgroup of animals who were moved from enriched to non-enriched housing or from non-enriched to enriched housing right after the first assessment. And if we do that, we find essentially the same picture here, which suggests that these measures, these individual differences are going to remain stable, are robust, even in animals who change, who are given new sets of life circumstances. So the same animal who is most stressed, for example, in the enriched housing is the same animal who's most stressed when his group moves to non-enriched housing. We had a second complementary welfare indicator in this project, which was a human approach test. Essentially, here piglets are led one by one into a novel arena, which is an empty pen, and a human enters and sits in the corner quietly for 3 minutes without moving. And during this period, we measure approach behavior and a variety of other behaviors exhibited by the piglet. Then we have a phase of approach by the human, where the human essentially creeps in a standardized manner towards the piglet to try to go touch them, and we see if the piglet allows themselves to be approached. I won't get so much into the statistical analysis of this. Safe to say that the outcomes of these tests were quite stable across time as well. So if we look at in this case, we tested it 3 times, at the start of the study, middle, and at the end of the nursery period. And if we compare the start and the middle, we have quite stable differences. If we compare the middle versus the end, whoops, once again, we have quite stable differences. And if we look at the robustness, again, in just a subsample of animals who move from enriched to non-enriched housing or vice versa, we again have differences that look quite stable. So what can we conclude from this first study? First, we have a couple of complementary welfare indicators that suggest that we have individual differences that are stable across time and robust to changes in housing, in this case, the start or the end of an enrichment treatment. However, sadly, we couldn't really get our principal welfare indicator to work. We didn't manage to get automated measures of judgment bias. I think this is something that would be worth trying again, but likely in older animals who are at a more mature state of cognitive development and who have longer periods in which we could train and test them. And especially it would be important to physically protect the animals during the test so they're not always being interfered with. I think sows would be a good target for this, given that at certain stages of their production cycle, they are housed singly, so there's no room for interference. And at other stages of their production cycle, in group gestation, they tend to have access to automated sow feeders, which again, physically protect them while they are in there. So this could be a nice context for testing them. And I think that if we get more and more evidence that these differences are stable across time and especially robust across types of housing, and we want to think about selection, this is necessarily going to require large quantities of data and efficient automated behavioral phenotyping. So that first project gives us some evidence, at least for some indicators, of stable and robust differences. And these, remember, are just based on spontaneous differences that exist between individuals. In our second project, we were curious about whether we could intervene on the animal's development to create some of these differences, to shape these differences. And we were interested in early life positive experiences that these animals can have. Specifically, we're interested in play. And the primary hypothesis that we want to test in pigs is that the function of play is to train animals to cope with unexpected, dangerous situations. This is a hypothesis that I think you can understand if you look at the pictures on the screen and realize that these animals who are playing look very silly. They're doing things like jumping off of logs onto each other. If you look at the sheep in the middle, they're twisting their bodies in the air and they're twisting their heads. So they're off balance and they can't even see where they're going to land. This rat on the right is pinning his buddy to the floor with all four paws on top of the buddy. So as soon as buddy moves, that rat is going to tumble to the ground. And the idea here is that they're doing this. They're getting themselves into these precarious situations where they'll lose their balance, they'll fall down, they'll be surprised by their opponent. They're doing it on purpose because this is going to teach them how to react in this kind of situation. This is how they're going to learn how to right themselves, get back on their feet, for example. And this will give them physical preparation for how to deal with this kind of event, but also emotional preparation, because they will have lived several times in a safe context through something like a real dangerous event that might happen later on in their lives. There is some evidence for this from other species. I'm showing you here something for a wild animal, which is the California ground squirrel. I think it illustrates the idea quite well. This is a species in which researchers, excuse me. This is a species in which researchers observed animals playing in the wild, and they trapped them and then performed some standardized assays on them in novel arenas and then let them go again. And they checked to see whether the play that they had observed was related to how the animals reacted in these novel arenas. And the test on the left was a test conducted in a novel arena that was empty, except they had added branches and soil and things like that. And they really wanted to see how bold each individual is, how much will this individual go and explore the crevices of the novel arena versus just staying in a corner or hiding. And what they find is that the animals who had played the most in the period before this test showed the most bold and exploratory temperament or personality. Meanwhile, on the right, this was a slightly different novel arena, which had branches and that kind of thing arranged in such a way that the animals could climb up them very acrobatically and leap out of the arena and run back into the wilderness. And so here they measured how quickly the animals could get out of this situation. And again, the animals who had played most in the intervening period were the ones who were able to get out of this situation fast. Put these two together, what does that tell us? That says that the animals who were the most playful were the ones who were least afraid of getting into a potentially risky situation by exploring that arena. And they were the ones who were most able to get themselves out of a risky situation. We can think about how this might be useful for a captive animal like a pig. Think about a stressor like transport to slaughter, for example. We're asking the animal to walk down a corridor, up a ramp, into a truck, which the animal maybe has never done before in its life. If we have animals that are less scared of doing this, they might be less stressed by this event. And then we're imposing some sort of physical challenge where they're being driven along a bumpy road, being bumped into by their cage mates, not cage mates, but by the other animals they're being transported with, and they might be better able to withstand this part of the event itself because of their physical competence. So that's where the interest for this project comes in. And this project is run by a doctoral student named Azizdine Shibai. And Azizdine is interested in looking, first of all, at an experimental approach, looking at whether we can give animals housing treatments that are meant to stimulate play. And these are, once again, environmental enrichment treatments, to see whether these would lead to decreased reactivity in stressful situations, stressful challenges that we will expose the animals to in the nursery stage. And because Azizdine is interested in looking at the long-lasting effects of early enrichment, as well as at the current effects or the effects of enrichment at the time when the challenges happen, he used enrichment treatments at the sow unit, so before weaning, in the form of extra space and manipulable objects, and then again in the nursery stage itself, where the challenges are taking place. The stressful challenges we used at about 4 and 6 weeks, twice each, and I'll explain why twice each in a little bit, are number 1, a transport simulation test, and number 2, an isolation test. And these are a little bit different. The transport simulation test is more of a physical challenge for the animals. The isolation test is not very physical at all, but something stressful for a gregarious species like the pig. We're using a transport simulation test because really we would have liked to just transport the pigs twice, but there are huge biosecurity issues involved in taking pigs out of the farm, moving them around in a truck, and bringing them back. So that was not allowed, unfortunately. So the way that we tried to simulate this as best we could within the building was by taking pigs from the room where they were housed, which is this yellow room number 3, and then moving them by leading them through the corridors over to the warehouse, which is number 1 over here in red. When they got to the end of the corridor and we were moving all the pen mates together, so these were once again in groups of 4 familiar animals, they got to this ramp, which is the same ramp that is used at this facility to send market weight pigs into the trucks that bring them to the slaughterhouse. So the piglets climbed up the ramp, and at the top of the ramp, they went into this cage, which is actually a repurposed metabolic cage that is generally used with bigger animals. And the fact that the bars of this cage are movable, can be fixed in any position, allowed us to, to adjust the space available to mimic commercial stocking densities during transport. Once the pigs were in the cage, we then had them pushed around inside the warehouse in a standardized fashion over a 40-minute period. So every 5 minutes, they would go through 1 minute of pushing, tried to get everyone to push at the same speed and with the same roughness as best we could. And we're taking data here behaviorally from cameras at the moment of loading, looking at when they climb the ramp, do they turn around, do they stop, do they climb over each other, and then also within the cage, in between the movements when we push them. We also took physiological measures, the first being salivary cortisol, first before the test as a baseline measure, and then after the test to see how stressful this event was. And we also took infrared thermography from different zones of the body, once again, to try to evaluate stress. The imagery from thermography is still under analysis, so I won't present any data from that part today. I'm going to tell you right away about the second test, which is a little bit simpler. The isolation test essentially involves leading the animal into an empty pen, which is in a separate room. This room number 2 here, which was completely empty at the time of testing. And so the animal is left in here for 3 minutes and is completely isolated physically and also auditorily from its own pen mates, which are in the adjacent room. And then we video the animal for 3 minutes. We also record vocalizations and we send them back home. So here we're taking behavioral measures and once again, thermography, which we will not present today. So let's recall our objective was to see whether the enrichment treatments pre-weaning or post-weaning would lead to decreased reactivity or stress in these tests. I don't have time to present the data in detail, but suffice it to say we had only some pretty minor treatment effects, and they were not really consistent in a direction that would allow us to say that enrichment gives the animal some sort of protection against these later stressful challenges. To make the link back to the first project, it is interesting to note that some of these measures were stable across time, given that we ran 2 repetitions of these tests, especially salivary cortisol after the transport was consistent across time, and behavior in the isolation test was very, very consistent. The second objective of this project is a little bit different. This is an additional objective where we're not trying to look at the effects of the treatments themselves, but more specifically at the role of play in the development of temperament or reactivity to these stressful challenges. So this is a correlational approach where we want to look at differences between individuals and differences between pens in how much play is going on to see whether that predicts how they will react to challenges. Are playful animals braver? Are they less stressed by the challenges we give them? And so as Edzin currently is observing video to quantify these differences between pens and between individuals. So we don't have the data on this yet, but I just want to point out something methodological here. Suppose that we do find that the piglets who play a lot are brave in these challenges. Would that mean that they became brave because they were playing a lot? Not necessarily. Maybe they were just braver to begin with, and braver animals play more. Maybe because they're spending less time looking around at potential threats. So how do we actually tease apart the causality here? The way I think we can do it is by being inspired by a certain lab in California, the Scott Nunez lab. This is the only lab I see using this paradigm aside from us now, and I think it's a really good paradigm, so I'm going to explain it. What they do is they don't do this. They don't observe play and then test the animals and try to say that whatever they're seeing, whatever differences they're seeing in the test are attributable to play, because the differences could have been pre-existing. Instead, they use the paradigm where they test the animals twice. So they run a first test, then they observe play in the intervening period. then they look at a second test. And instead of trying to correlate play behavior to behavior in the second test, they look for a correlation between play and the difference between test 1 and test 2. And then that means if they do have a positive correlation, which is actually what I showed you in the previous ground squirrel example, then they know that if these animals are braver in test 2, it's not because they were already braver in test 1, because they've controlled for that. So that's not explaining the difference in play, and more likely the difference in play is explaining the evolution of these animals' behavior over time. And so that's what we're trying to replicate here by concentrating Azzedin's behavioral observations of play in that period between the two tests. And he's going to be correlating play to change from test 1 to test 2. And those results, of course, are pending. So to conclude for this part of the project, we don't have any evidence so far that housing treatments that are meant to stimulate play reduce reactivity under stressful challenge. It does seem like individual differences are somewhat stable over time in the tests that we used here, and we are waiting on further results for play. Before telling you about our last project, I just want to mention that alongside Azzedin's regular data collection, we're also running a project where we're trying to develop automated methods to detect play in piglets. So we have accelerometers that were conceived or designed for us by colleagues in electrical engineering, which are small and lightweight. And so we can install them on the ears or on the legs of piglets. And they generate output that looks like this, with large spikes representing forceful movements that the animals are making. And we're going to be, well, right now we have a couple of undergrads who are trawling through videos like this to identify instances of social play, locomotor play, and object play. And we're going to see if we can find ways of detecting play based on the accelerometer outputs, which is an approach that has been used in dairy cattle already, dairy calves, and seems quite successful. Parallel to this, I co-supervise a student, Safine Gobibinbe, who is a computer vision whiz, and she has adapted a software for us called Track Anything so that we can efficiently do computer vision-based tracking of piglets in their environment and extract measures of it, like estimates of velocity and of position over time, relative position of the piglets. And we'll see if that, instead of the accelerometer data, or in addition to it, might be a good tool for automated detection of play. So hopefully maybe in about a year, we'll have results from this project in particular. Okay. Try to go a little bit fast through our last project, our last determinant of robustness. Here, we're not going to be talking about specifically a stressful challenge like a transport simulation test. We're going to be talking about a different kind of challenge, which is a challenge to physical health. And we're interested in the role of social dynamics in protecting against that kind of challenge. We know from research on human health, especially, also on lab rodents, there's not as much that's been done on farm animals. We know that social integration, meaning having positive social relationships with other individuals, engaging in affiliative behaviors with other individuals, is protective against a range of different disease outcomes. And social isolation is a risk. Here I have an example from humans that concerns cancer. But as I mentioned, this applies to a range of different disease areas, including infectious disease, which, as I mentioned earlier, is a big concern in the swine sector. And in particular, respiratory disease accounts for more than 50% of mortality in the nursery stage. It's the biggest risk to the piglets' health. And it accounts, if you take the whole post-weaning period all the way to slaughter, for something like 60 or 70% of all post-weaning mortality. The CDPQ, the Pork Development Center that I mentioned earlier, has something that they ran from 2015 up until recently in 2024 when they discontinued it, called the Natural Disease Model. And this is basically a response to this priority of the swine sector, this priority for taking care of this infectious disease problem. And they were essentially running this to look at genetic or genomic factors associated with resilience or resistance against disease. I'm going to describe how this model works. At the experimental CDPQ nursery, there are 2 zones and 2 phases through which the animals go. After weaning at the age of 3 weeks, they arrive in the quarantine area in which they stay for 2 weeks. And this is maintained with a high degree of sanitary precautions. So the animals are not exposed to pathogens in this area normally. But then at the age of 5 weeks, they move into what's called the challenge nursery. And here they undergo a sanitary challenge. The challenge nursery is deliberately maintained in poor sanitary conditions. So they know that there are pathogens circulating in this environment. And when a new batch of piglets arrives here, the previous batch of older piglets is still there. So there's a period of time where they share the same room, they share the same air. Some of the pathogens are transmitted. The older piglets leave, a new batch arrives later, and the pathogen transmission continues. In here, they have some of the main pathogens that affect pigs in commercial production, including porcine reproductive and respiratory syndrome virus, swine influenza, salmonella, streptococcus suis, and so on. And these kind of this pathogenic cocktail that causes mainly respiratory symptoms, which, as I mentioned, mimics the real types of symptoms we do see in commercial production. Obviously, this is a model that is not great for the welfare of the animals involved. And I did hesitate before deciding to run a project here, but I think it was worth investigating, aside from genetic factors that are involved in resilience, looking at other factors such as social behavior as potential protective effects, as something that could potentially provide provide protection against infectious disease in pigs, especially in a sector where biosecurity is such a huge concern and where so many animals are concerned. So this model gave us an interesting context in which we could study these social effects. And we had two objectives here, which somewhat mirror the objectives of the previous project. The first is to look at the effects of housing or husbandry treatments that we know from previous literature improve social dynamics within piglet groups to see if they also confer resistance against disease. And then the second was to characterize more closely the relationship between these social dynamics as we observe them and resistance to infectious disease. So the treatments we applied based on previous literature are, first of all, pre-weaning mixing, which you may also know as early socialization. This is a treatment in which piglets pre-weaning at the sow unit are allowed to mix with the individuals of a different litter from an early age, in our case at the age of 5 days. And this allows them to develop social competence early rather than have their first encounter with strangers be at the moment of weaning at arrival of the nursery. The second treatment, you're getting used to this, is environmental enrichment. And both of these treatments, we know from previous literature, can reduce negative interactions, so reduce the amount of aggression between the piglets. We know less about what they'll do to positive social interactions, nose-to-nose contacts, social play, and so on, which are also of interest for this project. The experimental design, briefly, is that we used 4 batches, and within each batch, there are 4 groups of 10 piglets. Half of these groups had been socialized at the sow unit, half of them had not been socialized. And then within each of these groups, this is a factorial design, within each of these groups, half of them got enrichment at the nursery, and half of them did not get enrichment. The enrichments were applied right away from the age of 3 weeks when they arrived in the quarantine area, and then they were maintained from the age of 5 to 10 weeks in the challenge area. And here you can see the pens are of unequal sizes. So the enriched treatments had not only manipulable objects, but also 50% more space than the standard treatments, which are housed here in the middle. So let's look at a few clinical outcomes. Here I've graphed the worst health score that each piglet received over the span of the 5 weeks. They're scored according to a grid developed by veterinarians every week. So, and I've extracted the worst score for each piglet. Let's look first at control piglets, the ones who were neither socialized nor enriched. About 15% of them died, which is a lot. I want to reassure you right away, these are not just piglets who are dropping dead. There were endpoints in place that could lead to euthanasia. Another 10% developed severe symptoms, and then the rest were about half moderate and about half mild symptoms at worst. If we look at the socialized group and the enriched group, now we're down to about 10% mortality, and we have an increasing number of animals in the mild category. And finally, in the group of socialized and enriched piglets, here we have no mortality. We have one kind of dark red one, which was classed very severe, but who recovered with treatment. And we have almost half the animals in the mild category compared to just over a third in the control treatment. We can also look at this in terms of the number of animals per pen, or the percentage of animals per pen who required medical treatment, who got sick enough to require individualized treatment. And again, we see the same pattern with significant effects of socialization and of enrichment. We also had a few effects on production or performance measures like food consumption, weight gain, but I'm going to gloss over, I'm going to skip those just in the interest of time. To look at our second objective, and I'm going to mostly skip this as well, we had 2 sources of behavioral data that would allow us to quantify the social behavior exhibited by these animals. One source is simply video and observing affiliative and aggressive interactions. And the other source of data is sensors worn on the animal's ear tag, which provide us with location data, much as has been used in the Finnish Hockey League, for example, because these are a Finnish product, and also accelerometry. And so this lets us do things like take estimates of where the animals are at any given time. You can see the mapping of the coordinates onto a perspective distorted video is not great, but you can see some of these dots moving along with the pigs that they represent. And we can derive estimates of proportion of time spent active from the acceleration measures or from the velocity measures. So this is an example here showing that we see nice diurnal rhythms, and then we can extract individual differences on piglets to use as an outcome measure of sickness behavior. Piglets who become very inactive during the period of peak clinical severity. And we can also use this to estimate things like social lying behavior. We have animals who choose to be inactive with each other. And we are now going through analyses. I won't present results, but social network-based analyses based on these measures. And we see very consistent differences in terms of who spends a lot of time lying with each other, who avoids lying with other pigs, and we're going to see whether that predicts disease outcomes. So what we can conclude so far from this project is that we have some indicators of higher robustness, especially in terms of clinical outcomes associated with socialization and enrichment, and the rest of our analysis is largely ongoing. So I'm going to end with just 2 minutes about the concept of robustness and why I think it's important. We can think about how farming has changed through time. I think robustness is important because farming methods are always changing. So I've illustrated it here for sows. Over on the right is the way that we typically, one version of the way we typically keep sows or are moving towards keeping sows during gestation in groups indoors. If we back up a few years, it looked like this. If we back up even further, it looked something more like this. And now the question, of course, is, well, which animal is best suited to overcome the challenges in each of these environments and to maintain good welfare? The answer is it's probably not the same animal because the challenges are not the same. The problem comes when we imagine what's going to happen in the future. I don't know what swine farms will look like in 30, 40, 50 years. I don't think anyone truly does. And so it's hard to project ourselves into the future and think, what should the animal look like who's going to be well suited to the specific challenges of that environment? And so that's where the risk comes in, I think, if we try to shape animals to a specific environment, that we create animals who are specialists or well suited for that environment, but not necessarily well suited to maintain good welfare in an alternative environment, which is unpredictable. And that's where I think the focus on robustness is very interesting, in the sense that it could offer us a way to try to figure out how to promote robustness in animals, how to promote good welfare across a range of different environments, a range of different living circumstances. I put that black bar all the way at the top. Maybe that's optimistic, but we could have a bar that's relatively higher than the others. Okay, I want to end by thanking all the graduate students I work with who are pictured here, mentors and collaborators on the left, undergraduates on the right, and all of our funders and partners who have their logos displayed. And also, I've been asked to do a little bit of promo, December 4th, 12:30 Eastern, Zoom link on the screen. Do not miss the CCSAW mini-series Animal Law Talk. Thank you very much. I'd be happy to answer any questions and discuss.